Welcome. Today, we're diving into the principles, workings, and applications of NMR spectroscopy. This technique is a powerful tool that helps us understand the structure and behavior of molecules. We'll explore how it works and why it's so valuable across science and industry. Let's get started. Let's talk about NMR spectroscopy. It's a powerful tool we use to figure out the structure of molecules. How? By measuring the magnetic behavior of certain atomic nuclei, like hydrogen-1 or carbon-13, when they're inside a strong magnetic field. This technique gives us a detailed fingerprint of the compound's architecture, helping us understand what it's made of and how its atoms are connected. It's like a molecular detective story, told through magnetism. NMR, or nuclear magnetic resonance, is a powerful tool that gives us insight into molecules in several ways. It reveals molecular structure, how atoms connect and arrange themselves. It tells us about the chemical environment, showing how atoms influence each other. It also helps us understand molecular interactions and the dynamics, how molecules move and change over time. Common nuclei we study with NMR include hydrogen-1, carbon-13, and phosphorus-31, each offering unique information. Let's talk about NMR, or nuclear magnetic resonance spectroscopy. It works by exposing certain atomic nuclei to radiofrequency radiation while they're inside a strong magnetic field. Some nuclei, like hydrogen-1, carbon-13, fluorine-19, and phosphorus-31, have something called nuclear spin. Because these nuclei are charged and spinning, they act like tiny magnets, each with its own magnetic moment. Think of a hydrogen nucleus. Its spin turns it into a miniature magnet, ready to interact with the magnetic field around it. Now, let's talk about what happens when we put a sample into a strong magnetic field, which we call B0. The tiny magnetic moments inside the nuclei start to line up with this field. For a simple nucleus like hydrogen, there are just two energy states, one where the spin aligns with the field, which is lower energy, and one where it opposes the field, which is higher energy. This magnetic field creates an energy gap between these two states. That difference is key to what comes next. Now, we're at the point where radiofrequency radiation is applied to the sample. When the frequency of this RF pulse matches the exact energy gap between two spin states, resonance happens. This means the nucleus absorbs the energy and jumps from a lower energy state to a higher one. The key relationship here is that the energy difference equals Planck's constant times the frequency of the radiation. It's this precise match that makes the whole process work. Once the RF pulse stops, the excited nuclei begin to settle back to their original state. This process is known as relaxation. As they relax, the nuclei emit a faint electromagnetic signal. It's this very signal that the NMR instrument picks up and uses to create the images or data we need. So, relaxation is key. It's how the system tells us what's going on inside. Once the signal is detected, it's sent to a computer for processing. This creates what we call an NMR spectrum, a kind of molecular fingerprint. The position of each signal, known as the chemical shift, reveals the chemical environment around the atoms. Integration shows us how many protons are present relative to each other, and splitting patterns tell us about neighboring nuclei, giving us a deeper look at the molecule's structure. Together, these details unlock the story hidden in the spectrum. Here, we see a diagram of an NMR spectrometer, the heart of nuclear magnetic resonance analysis. This instrument uses powerful magnets and radio waves to reveal the structure of molecules. Each part plays a key role, from the magnet that aligns atomic nuclei to the detector that captures their signals. Understanding this setup helps us grasp how NMR turns invisible molecular details into clear, usable data. It's the foundation for everything we'll explore next. Let's walk through how NMR works, step by step. First, we place the sample inside a tube, then into a strong magnetic field. The nuclei inside start to align with that field. Next, we apply a radiofrequency pulse, which excites the nuclei, making them absorb energy. Afterward, they relax back to their original state, releasing energy as they do. This released energy creates an electrical signal, which a computer processes to generate the NMR spectrum we analyze. Let's break down the basic parts of an NMR spectrometer. At its core, there's a strong magnet. That's what creates the magnetic field we need. Then, we have the sample tube, where the material we're analyzing sits. The probe surrounds the sample, sending and receiving signals. The RF transmitter sends radio waves to excite the sample, while the RF receiver picks up the response. Shim coils fine-tune the magnetic field. The lock system keeps everything stable. And finally, the computer processes all the data we collect. Let's talk about the heart of the NMR instrument, the magnet. This component is absolutely crucial. Its job is to create a powerful magnetic field, one strong enough to align the tiny nuclear magnetic moments inside the sample. Without this alignment, resonance simply wouldn't happen. Today's instruments usually rely on superconducting magnets to achieve these high field strengths. And we measure that strength in Tesla, a unit that tells us just how intense the magnetic field really is. Next, let's look at the sample tube and the probe, two key players in this process. The sample tube is usually a narrow glass tube holding the sample dissolved in a solvent. It's important that the sample is pure and concentrated enough for clear results. Then there's the probe, which sits inside the magnet. It has radio frequency coils that send pulses to the sample and pick up the signals coming back. Together, they make the measurement possible. Now, let's talk about the RF transmitter. This component generates the radio frequency pulses we need. These pulses deliver energy to the nuclei, flipping their spin state. It's crucial that the frequency matches the resonance condition of the specific nucleus we're studying. In simple terms, the RF transmitter sends the right signals at the right frequency to make the nuclei respond, which is the first step in capturing our sample's data. After we excite the nuclei, they start to relax back to their original state, and in doing so, they emit a faint electromagnetic signal. That's where the RF receiver comes in. Its job is to pick up this weak NMR signal, convert it into an electrical signal, and then send it off to the computer for analysis. It's important to remember, the transmitter sends out the RF energy, while the receiver is the one that listens for the NMR signal. They work as a team to capture the data we need. Now, let's talk about shim coils and the lock system. Shim coils are crucial because they make the magnetic field highly uniform and homogeneous. Why does that matter? A uniform field gives us sharper, better resolved peaks, which means clearer data. Then there's the lock system. It keeps the magnetic field stable throughout the experiment. Without that stability, our results could drift or become unreliable. Together, these components ensure precision and consistency in what we measure. Now let's look at the role of the computer in NMR. It starts by collecting the signal, the raw data coming from the experiment. Then it processes this data, turning what we detect into something meaningful. Specifically, it converts the time domain signal, known as the free induction decay, or FID, into an NMR spectrum. This transformation happens through a mathematical tool called the Fourier transform. Finally, the computer displays and stores the spectrum for us to analyze. Let's walk through the complete process behind nuclear magnetic resonance, step by step. It all starts with the sample placed in a strong magnetic field, which aligns the nuclear spins inside. Then, we apply a radio frequency pulse, that's the RF pulse, which excites those spins. After excitation, the nuclei relax back to their original state, creating the NMR signal. This signal is picked up by the receiver, sent to the computer, and finally processed into the NMR spectrum we analyze. This flow captures the full journey from sample to spectrum. An NMR spectrum is a powerful tool for uncovering molecular structure. First, the number of signals tells us how many distinct nuclear environments are present. Then, the chemical shift reveals details about those environments themselves. Integration shows us the relative number of protons in proton NMR, while splitting patterns give clues about neighboring nuclei. When we put all these pieces together, we get a clearer picture of the molecule's architecture. Let's talk about where this technology really shines, its applications. In metabolomics, it can analyze a wide range of samples, from blood and urine to cell and tissue extracts, even other biological fluids. This opens doors for metabolic profiling, biomarker research, disease studies, and mapping out metabolic pathways. On the protein side, NMR helps us understand protein structure, how proteins fold, the changes they undergo, and how they interact with other molecules. It's a powerful tool across multiple fields. NMR plays a crucial role in studying natural products, things like alkaloids, flavonoids, terpenoids, and other bioactive compounds. It helps us figure out their molecular structure, identify functional groups, and understand how atoms connect. Sometimes it even reveals stereochemical details. In drug discovery, NMR lets us explore how drug molecules interact with proteins, showing us if and where binding happens, and how the protein changes afterward. It's a powerful tool for uncovering molecular interactions that matter. Let's look at how NMR plays a crucial role in biotechnology and pharmaceutical research. It helps us characterize pharmaceutical compounds, ensuring we understand their structure. We use it for quality control, keeping products consistent and safe. It's invaluable for analyzing metabolites and monitoring fermentation processes. Beyond that, NMR tracks bioprocesses and explores how biomolecules interact. And when we face unknown compounds, it's our go-to tool for identification. In short, NMR is a versatile powerhouse in this field. As we wrap up, it's clear that NMR offers powerful advantages. It's non-destructive, gives detailed structural insights, and reveals molecular dynamics with minimal sample prep. But it's not without challenges. The instruments are costly, spectra can get complex, and interpreting results demands expertise. Balancing these strengths and limits helps us use NMR to its fullest potential.